What is life really like before the emergence of the first animals? Dear traveler, greetings. Today, we look much further back than the Cambrian and Ediacaran to a forgotten world that existed even before the emergence of the first animals. From silent oceans, where immense carpets of microbes stretched, to reefs built by stromatolites, life was reduced to tiny but essential forms. A discrete reign, and yet decisive, for it was there that oxygen, complex cells, and the earliest cooperations of life were invented. But how did this world of microbes give way to the first animal creatures? But before we embark on a new adventure, don't forget to like the video and subscribe to the channel to miss nothing. Thank you and bon voyage. About 3.5 billion years ago, shallow marine margins formed carbonate platforms bathed by warm waters. Nearby, evaporitic zones developed under intense evaporation, depositing salts and minerals. In this setting, tides and heat favored the formation of laminated sediments colonized by biofilms, communities of microorganisms producing a sticky substance that trapped particles in successive layers. In this habitat, methanogenic archaea, members of the eukaryotic, transformed carbon dioxide and dihydrogen into methane. This methanogenesis, active in oxygen-poor sediments, provided them with an environment without real competition. The produced methane strengthened the greenhouse effect and maintained a warm climate. Thus, for millions of years, these invisible microorganisms quietly shaped the Earth's environment. On the sunlit surface of the sediments, other bacteria played a key role. These anoxygenic phototrophs used light as energy without producing oxygen. Unlike later cyanobacteria, they exploited ferrous ion or hydrogen sulfide rather than water. The chlorobi and chloroflexi thrived even in low light, while some proteobacteria occupied various niches adapted to variations in light and chemistry. Over time, the interaction of microorganisms and sediments shaped structures still visible in ancient rocks. The most famous are stromatolites, of laminoid or conical shape. They result from microbial mats that secrete a sticky matrix, trap particles, and then rise by stacking layers. Notable fossils exist at Strelley Pool and the Dresser Formation in Australia, among the oldest evidence of life. We also observe MIS, sedimentary structures induced by microbial activity, with varied patterns, ridges, cracks, or ripples. To understand this era, one must examine the chemistry of the oceans and the atmosphere. The seas, rich in dissolved iron in the form of ferrous ion, were called ferruginous. In a nearly oxygen-free world, this iron remained in solution. The atmosphere, saturated with methane, warmed the surface despite a less luminous sun. On the shallow margins, water commonly reached over 30 degrees, sometimes exceeding 50 near volcanoes. This thermal and chemical setting favored the diversification of metabolic strategies of archaea and bacteria. Among these strategies, methanogenesis converted carbon dioxide and dihydrogen into methane in anoxic sediments. This gas ascended to the water or the atmosphere, strengthening the greenhouse effect and partially filtering solar radiation, helping life adapt to intense light. Thus, this oxygen-poor, methane-rich atmosphere shaped strange landscapes, often enveloped in mist, depending on water and temperature. Other microorganisms exploited different electron sources for their anoxygenic photosynthesis. Photoferrotrophic bacteria used light to oxidize ferrous ion into insoluble ferric compounds, forming iron bands in the sediments. The iron, associated with silica or other elements, deposited in successive layers, tracing patterns witnessing distinct sedimentation episodes. Although partially abiotic, this process was greatly accelerated by microbial action. For some bacteria, hydrogen sulfide was a key resource, which they oxidized into sulfur or sulfate in a sulfurous photosynthesis without producing oxygen. Adapted to transitional zones, they thrived where light persisted, but oxygen was lacking. This primitive soup of iron, sulfur, and carbon reflected the chemical richness of ancient seas, where an astonishing variety of microbial processes unfolded. Imagine shallow seashores, bathed in light sometimes filtered by particles. At the bottom, methanogenic archaea produce a constant flow of methane. A few centimeters above, anoxygenic chlorobi, chloroflexi, and proteobacteria capture light, fix carbon, and release sulfur or iron oxides. In some places, these microorganisms form small mounds, which become stromatolites. The tides bring nutrients and salts, while the heat extends the evaporitic expanses, leaving traces of crystallized minerals. In volcanic regions, geothermal activity brought up superheated fluids, enriching the environment with nutrients and ferrous ions. Temperatures sometimes exceeded 60 degrees, only allowing thermophiles to survive. This microbial diversity fostered evolutionary innovations. Genes encoding heat-resistant enzymes were transferred between species, enabling several lineages to colonize different coastal sites of primitive Earth. The biofilms of the time were more than just slippery mats. Indeed, they offered a collective way of life, with varied microenvironments. The upper illuminated layers hosted phototrophs, while the depths housed communities managing sulfur or producing methane. As these films thickened, they developed vertical differentiations, creating several ecological niches and cementing sediments, laying the foundation for future durable geological structures. Beige. Mists are microbial imprints, visible on the surface as ripples or inside sediments in the form of cracks. For geologists, they reveal that a living film influenced the texture of the sediment, altering cohesion, water infiltration, and resistance to erosion. Each fossil pattern thus tells the story of the movements of microorganisms and the physico-chemical conditions that prevailed at the time. Iron played a central role in the early world, visible in banded iron formations. These strata alternate red, ochre, and gray colors, according to microbial activity and iron content. Oxidizing microorganisms quickly precipitated iron into insoluble oxide, while less active periods produced lighter, often siliceous layers. Over millions of years, this alternation created spectacular accumulations, evidence of a unique chemistry. Between 3.5 and 3.3 billion years ago, atmospheric oxygen was very rare, present only in localized pockets. The air was rich in methane, trapping heat and fostering microbial life. Methanogenic archaea reinforced carbon recycling, while phototrophic bacteria exploited ferrous ions, hydrogen sulfide, and other reduced molecules for their anoxygenic photosynthesis, thus consolidating their presence in various environments. The study of stromatolites and mists allows us to reconstruct this ancient narrative. Their internal structure often reveals an alternation of organic and mineral laminae, each cycle reflecting an ecological sequence. Microorganisms proliferated, secreted their substances, then were sometimes covered by new sediments. On a geological scale, these successive biofilms created reliefs that could reach several tens of centimeters, depending on the stability and duration of the construction episodes. The conversion of ferrous ion into iron oxide, whether abiotic or microbial, left its mark in the rock. The banded iron formations, sometimes thick, hundreds of meters, show that the primitive Earth was not a desert, but a world where chemistry and life interacted. Some layers reveal variations in oxidation, suggesting that the biosphere was already influencing the global circulation of iron. Reconstructing this distant past reveals the importance of a microbial biosphere. Archaea and bacteria, through biofilms, methanogenesis, photoferrotrophy, and sulfur photosynthesis, stabilized the climate, reshaped sediments, and initiated the iron cycle. Their actions governed the chemical balance of coastal margins and oceans. Laminates, stromatolites, and mists are today's witnesses of a young Earth, bathed in intense solar rays and iron-rich seas, where these ingenious microorganisms laid the groundwork for future life. Thanks to fossil clues and geochemical analysis, specialists continually discover new facets of these early communities. The ocean, rich in ferrous ions, offered a vast reservoir of energy. The metabolism of iron, carbon, and sulfur transformed the environment, altering the atmosphere and marine deposits. These microorganisms, weaving their mats and releasing methane, gradually paved the way for more diversified ecosystems. Between 3.2 and 2.8 billion years ago, the first oxygen oases appeared above coastal cyanobacteria mats. Imagine a warm shoreline, bathed by an iron-rich sea, with an atmosphere almost devoid of oxygen. Yet, these photosynthetic communities liberate dissolved oxygen locally, creating thin, oxygenated enclaves in a still predominantly anoxic world. The cyanobacteria, morphologically varied, include filamentous and coccoid forms. Filaments organize into bundles or sheets, while coccoids form gelatinous microcolonies, sometimes elevated in stromatolitic domes. These laminated mounds, alternating sediments and living films, form when microbial growth traps particles and triggers mineralization. Over time, these stromatolites can reach several tens of centimeters, a remarkable feat for microscopic organisms. Near the surface, the oxygen released by photosynthesis reacts with dissolved iron, transforming it into oxyhydroxides and creating reddish deposits. This chemistry indicates localized oxygen and imposes oxidative stress on anoxic bacteria and archaea. Over just a few millimeters, gradients are established. Above, there is a thin, oxygen-rich veil. Below, an area soaked in hydrogen sulfide. These discontinuities, although tiny, play a crucial role in this primitive world. For anoxic microorganisms, oxygen becomes threatening as it generates reactive derivatives like hydrogen peroxide or free radicals, damaging membranes and proteins. Some develop catalysts or peroxidase enzymes for protection, while others migrate to oxygen-free zones or take refuge in deep sediments. This coexistence of photosynthetic producers and strictly anoxic organisms already creates spatial segregation. These oxygen oases favor the emergence of microaerophilic microorganisms. They position themselves in the thin layer where oxygen is sufficient for their metabolism while avoiding toxic radicals. This strategy allows them to exploit the organic matter of cyanobacteria and ferric minerals while developing new cellular protection mechanisms suited to this partially oxygenated environment. The day-night alternation creates cycles of oxygenation and deoxygenation. Intense daytime photosynthesis increases dissolved oxygen around the mats, while nighttime sees it diminish, allowing anoxic communities to thrive. This daily back and forth forces microorganisms to adapt. Photosynthetics store reserves, and anoxics take advantage of nighttime periods to grow. Thus, niche distribution is structured, and microbial diversification is stimulated. The hydrogen sulfide gradient between the oxygenated and anoxic zones creates a precise chemical stratification. Cyanobacteria occupy the surface, microaerophiles remain just below, and methanogenic archaea, along with sulfate-reducing bacteria, thrive in the deep sediments. Each stratum exploits its microenvironment, forming a mosaic of microuniverses where astonishing metabolic innovations express themselves at a millimeter scale. Oxidative stress highlights the crucial role of catalyses and peroxidases, which break down hydrogen peroxide and neutralize free radicals. These reactions protect microorganisms even at low oxygen levels and foreshadow the evolution of aerobic respiration, which will later exploit oxygen to produce more energy. Thus, from this early stage, a defense mechanism becomes a fundamental metabolic advantage. The diurnal cycles of oxygenation and deoxygenation regulate the formation of stromatolites. Oxygen alters the solubility of minerals, creating thin films on biofilms that can disappear or be covered at night. Day by day, these alternations stack laminations, slowly growing the domes. Millions of years later, these structures become geological archives, allowing scientists to reconstruct the first oxygenating ecosystems. The first Archean oxygen oases, limited but significant, testify to the complex interactions between microorganisms and environmental chemistry. Cyanobacteria produce a local flux of oxygen, forcing the anaerobics to seek refuge or adapt. On a millimeter scale, the oxygen-hydrogen sulfide gradient imposes a subtle balance of adaptations, creating a mosaic of habitats and initiating the gradual colonization of varied environments through increasingly ingenious strategies. The presence of rare but crucial oxygen favors microaerophiles and the enhancement of their enzymatic defenses. The day-night cycles structure activity windows and initiate regulation rhythmically influenced by light. In these coastal micro-landscapes, the precursors of planetary oxygenation, the future formation of the ozone layer, and later, the explosion of complex life forms take shape. These modest oxygenated niches mark the first spark of the oxygen revolution. Imagine Earth 2.8 to 2.5 billion years ago. The continents are barely emerging, and life remains microbial. The air, poor in oxygen and rich in methane, gradually forms a diffuse organic haze, a veil floating around the planet. In these ancient oceans, microorganisms adapt to this radically different world, laying the foundations of a chemical environment still far from our modern atmosphere. This organic haze forms by photolysis, where sunlight breaks down methane into more complex carbon chains suspended in the atmosphere. As they accumulate, they create a thick veil that increases the albedo, reflects some solar rays, and modifies the surface temperature. This fog also acts as a natural ultraviolet filter, partially protecting the biosphere from excessive radiation while shaping the environment of microorganisms. In this dim light, microbial life follows new balances. Methanogens produce methane from carbon dioxide and hydrogen, while methanotrophs consume this gas for their energy. Some develop in anaerobic environments by coupling methane oxidation to sulfate or iron reduction. Thus, these two groups create a permanent dance, balancing the production and consumption of methane in the environment. This interplay between methane production and consumption influences marine geochemistry. Part of the methane escapes into the atmosphere, fueling the organic haze, while the rest is oxidized by methanotrophs. In anoxic sediments, this oxidation couples with sulfate or iron reduction, maintaining balance. In these ancient seas, stromatolites of varied forms, columns, cones, or tufted mats, appear. These structures form when microbial mats, including cyanobacteria, trap and cement mineral particles. By the end of the Archean, the water chemistry and the distribution of ecological niches favor this morphological diversity, indicative of vertical differentiation and increasingly complex microbial organization. In stromatolites, each layer houses adapted microorganisms. Phototrophs thrive on the surface thanks to light, while deeper layers host anoxic communities exploiting other metabolic pathways. This vertical structuring creates chemical gradients and a complex architecture visible in the alternation of successive laminas, revealing how microscopic life organized and diversified its niches in these ancient mats. Stromatolitic cones form when central growth dominates the periphery, while other mats create pillars or tufted outgrowths. Each shape reflects local conditions, such as current, water chemistry, or suspended particles. These stromatolites thus become true geological records, revealing the presence of the methane haze. They illustrate how microorganisms, interacting with their environment, allowed life to persist on early Earth. The end of the Archean marks a turning point for the planet. Methanogens produce methane in anaerobic niches, while the first methanotrophs harness this gas through sulfate or iron. The organic haze formed by methane photolysis reflects sunlight and filters UV rays, shaping the appearance of the young Earth. In this ever-changing world, the balance between methane production and consumption remains vital for the biosphere. We are plunged approximately 2.43 to 2.32 billion years ago, during a pivotal time in Earth's history. The terrestrial landscapes are unrecognizable compared to today's. The coasts are mere strips of rock, devoid of vegetation, and the air has not yet attained its modern composition. This time period corresponds to the Great Oxidation event, a moment when the concentration of oxygen, previously almost nil, begins to increase, paving the way for the emergence of more complex life. Before this transition, the atmosphere is rich in methane, a powerful greenhouse gas, and the planet is bathed in a dense organic haze that filters sunlight, sometimes tinting the sky orange. Gradually, this haze dissipates, leaving space for the slow yet continuous accumulation of oxygen. To grasp the magnitude of this change, it is crucial to understand the notions of redox shift and chemical distribution in the ocean and atmosphere. The term redox denotes chemical reactions involving electron transfers between different elements, among which iron, sulfur, and carbon play a central role. Before the Great Oxidation event, significant amounts of iron remain dissolved in the oceans because oxygen is almost absent to oxidize it. This iron thus remains in a reduced, i.e., non-oxidized form. Once oxygen appears, even in low concentration, it reacts with the dissolved iron, transforming it into a more stable oxidized form and precipitating out of the water, profoundly altering the sedimentary deposits. The accumulation of these iron oxide-rich layers constitutes a true geological signature of this chemical shift. The disappearance of the methane haze results from a subtle chemical chain reaction related to the gradual accumulation of oxygen. As long as the methanogenic archaea produce methane and oxygen remains nearly absent, the haze persists. However, when cyanobacteria, capable of photosynthesis that releases oxygen, multiply in the warm, shallow coastal waters, oxygen production increases locally. This reactive oxygen then interacts with methane, reducing its concentration and causing the progressive dissipation of the organic veil. For an observer, the orangish sky left by the haze clears, revealing an atmosphere still primitive, but already evolving toward a more oxygenated environment. In this landscape, a major ecological upheaval is taking place. Cyanobacteria are the main beneficiaries. These microorganisms, often grouped in mats in coastal areas, now have a more open environment to grow and spread. Through their photosynthesis, they absorb carbon dioxide to produce organic matter while releasing oxygen. As their numbers increase, the local concentration of oxygen above their colonies also grows. This phenomenon contributes to a slow but irreversible oxygenation of surface waters, initiating the chemical transformation of the planet. In contrast, anoxygenic phototrophs, which derive their energy from molecules like hydrogen sulfide, experience a decline. Their metabolism is disrupted or even blocked by the presence of oxygen. Previously abundant, these phototrophs see their environment change. Light is still available, but oxygen introduces a chemical stress that is difficult to manage. Nevertheless, these organisms do not completely disappear. Some restricted, isolated basins, minimally stirred by currents, remain anoxic. In these refuges, anoxic life continues to thrive, sheltered from the progressive oxygenation of the oceans. This winners and losers phenomenon perfectly illustrates the evolution of the microbial world at this time. Cyanobacteria dominate in well-lit coastal areas, while sulfur bacteria and other anoxic organisms retreat to deeper waters or confined environments. Gradually, this dynamic shapes the distribution of different microbial groups on a global scale and sets the stage for major biological and chemical transformations, heralding future upheavals in the biosphere. Another key element of this period concerns sulfur. Geological records show the presence of a phenomenon called mass-independent fractionation of sulfur. Specifically, when sulfur undergoes photochemical reactions in an atmosphere very low in oxygen, its isotopes can exhibit unusual signatures that no longer follow the simple proportions related to their mass. This is called mass-independent fractionation because the distribution of isotopes escapes the classical rule. As soon as oxygen becomes more present in the atmosphere, these imprints gradually disappear. Oxidizing conditions modify the chemistry of sulfur and its transfer between air, ocean, and sediments. Sulfate, the oxidized form of sulfur, then spreads into the oceans, promoting the rise of microorganisms specialized in desulfation. These organisms exploit sulfate to produce energy and release hydrogen sulfide. This metabolic pathway, previously marginal, gradually becomes central as sulfate stabilizes in primitive seas. Geologically, the Earth's oxygenation event is also marked by microfossils. In sublittoral sediments near the coasts, spherical coccoids grouped in colonies and elongated filaments like fine microscopic hairs are observed. These microbes, preserved by the mineralization of sediments, testify to intense biological activity. The analysis of silicified cell walls allows us to distinguish between rounded and filamentous cyanobacteria or other photosynthetic bacteria. Their abundance often coincides with chemical clues suggesting the presence of oxygen, even at low concentrations in the water column. To a current observer, it may seem difficult to grasp the importance of unicellular organisms. Yet, these tiny forms of life, in mats or floating, shape Earth's chemistry during the oxygenation event. By capturing carbon dioxide, releasing oxygen, and triggering new metabolic reactions, they cause immense planetary changes. Namely, precipitation of dissolved iron in oxides, disappearance of sulfur's mass-independent isotopic signatures, and reorganization of aquatic trophic networks. These accumulated transformations set the stage for the evolution of subsequent eras. Before the rise of oxygen, Earth could not host complex animals that require a sufficient level of oxygen for their metabolism. This chemical and climatic transformation alters the Earth's surface. The conversion of methane to carbon dioxide by oxygen reorganizes the radiative balance, as methane, more potent than CO2 as a greenhouse gas, sees its concentration decrease, influencing global temperature. Geological and climatic reconstructions show that this mechanism played a role in climate stability, although the exact extent remains debated. On shallow marine margins, the retreat of sulfur phototroughs creates heterogeneous microenvironments. On the surface, cyanobacteria produce oxygen thanks to sunlight. Lower down, in less lit zones, anoxic microorganisms persist, exploiting sulfur, iron, or methane. This millimeter-scale stratification regulates metabolisms, restructures ocean chemistry, and promotes the formation of laminated sediments enriched in oxidized minerals. Thanks to their thick walls and sophisticated survival mechanisms, cyanobacteria colonize many ecological niches. Stromatolites, formed by the fixation of sediment particles in bacterial mats, multiply in certain sectors. The local accumulation of oxygen stabilizes sediment chemistry, iron oxidizes, and forms protective mineral crusts on or between microbial layers. These levels, preserved in rocks, offer precious evidence of the rise of oxygen and the ongoing chemical upheaval. As the methane haze disappears, solar radiation, particularly ultraviolet rays, reaches the surface more directly. Some microorganisms must then adapt to these new conditions. However, the gradual accumulation of dioxygen initiates the formation of a rudimentary ozone layer, capable of slowing down harmful ultraviolet rays. Although stable, this protection will take a long time to consolidate, with the first traces appearing alongside the rise of dioxygen. Anoxic refuges, far from the coasts or isolated by geological barriers, become sanctuaries for organisms thriving without dioxygen. In these confined environments, chemistry is based on sulfur and reduced iron. Sulfate reducers exploit sulfate, while others specialize in methanogenesis or anaerobic methanotrophy. This metabolic diversity, discrete but vital, supports ocean productivity while preserving ancient lineages unable to tolerate dioxygen. The analysis of more recent sediments shows the gradual decrease of sulfur mass. Independent fractionation signatures, these indices, common in ancient rocks, fade as soon as a certain amount of dioxygen appears. They allow us to date the beginning of the oxygenation event, fixed at about 2.43 billion years ago. At the end of this phase, dioxygen remains a minority, but its role in Earth's dynamics becomes already central. Microfossils, witnesses to this upheaval, illuminate life in sublittoral areas in shallow waters. Sedimentary layers preserve many spherical or filamentous remnants, sometimes in colonies. Their diversity increases with the rise of dioxygen, reflecting the progressive adaptation of microorganisms. The filaments form rafts of organic matter on the surface of sediments, while coccoids colonize microcavities and accumulate substances that mineralize. Silicification replaces organic matter with silica, freezing these forms and offering geologists today the opportunity to trace this global turning point. The Lomagundi excursion and the appearance of the nitrate effect, which occurred about 2.22 to to 2.06 billion years ago mark a major episode in Earth's history. At that time, bacteria and archaea already populated shallow waters. Environmental conditions changed significantly, causing upheaval in biochemical processes. The synthesis of organic matter from inorganic components experienced a spectacular boom. With this increase in productivity, the abundance of nutrients plays a crucial role, particularly nitrogen, essential for the synthesis of proteins and DNA. In suboxic or low oxygen environments, new processes develop. Indeed, nitrification transforms ammonium into nitrate, and denitrification returns nitrate to gaseous nitrogen. Thus, nitrogen circulates differently in the oceans, giving rise to the nitrate effect. It is characterized by an increase in available nitrate, favoring the growth of photosynthetic organisms. To grasp the nitrate effect, one must imagine a vast marine ecosystem where ammonium, nitrite, nitrate, and gaseous nitrogen circulate. In previous periods, nitrate was limited, hindering the growth of certain organisms. The rise of marine nitrate represents an ecological turning point. Cyanobacterial mats, forming biofilms on sediments or floating nearby, expand into new areas. Able to capture light and fix carbon dioxide, these microorganisms proliferate further and contribute to the overall enrichment of coastal ecosystems and continental shelves. The extension of cyanobacterial mats towards the open sea corresponds to their expansion beyond shallow coastal areas. Suboxic waters, with a low oxygen content, offer them a stable and favorable environment for growth. The formation of subsurface redox gradients, over a few centimeters in depth, creates zones of chemical transition. On the surface, oxygen allows cyanobacteria to thrive, while in depth, other microorganisms exploit the scarcity of this gas for their metabolisms. These redox gradients favored notable biological diversity. Each chemical zone hosted specialized species. On the surface, organisms practicing oxygenic photosynthesis released oxygen into the water. Just below, in more oxygen-poor conditions, microbes perform transformations like denitrification or the use of sulfur compounds. This stratification illustrates the intimate coevolution between chemistry and life, giving rise to varied ecological niches. During the Lomagundi excursion, the accumulation of nitrate in the waters particularly stimulated the growth of many species. On the paleontological level, numerous rocks derived from marine sediments preserve traces of life between about 2.22 and 2.06 billion years ago. Specialists have identified microfossils corresponding to particular microcolonies, such as Eoanthophysalis-like forms. These small coccoid structures, often grouped in gelatinous films, appeared in tiny protected cellular groups. This organization allowed them to survive in unstable environments, especially on the edges of sediments where water and chemistry could change rapidly. In the sediments of this period, spheroids of 5 to 50 and segmented filaments are found. The spheroids correspond to small rounded colonies, while the filaments form chains of juxtaposed cells. These morphologies testify to the already significant diversity of microorganisms during this interval, rich in biological innovations. Imagine a vast shoreline bathed by a shallow sea about 1.9 billion years ago. In these waters called the Paleoproterozoic Sea, one of the most fascinating fossil-bearing assemblages in the history of life was formed, the Gunflint Biota. The term biota refers to the array of life forms preserved in specific rocks. Here, it is about the famous Gunflint cherts, located around Lake Superior. Rich in silica, these rocks allowed exceptional preservation of microfossils, transforming these formations into true time capsules, containing unicellular organisms that lived in conditions hard to imagine today. To understand the importance of these discoveries, one must first place the facts in the geological and environmental context of the time. The Paleoproterozoic corresponds to an era of major changes, notably marked by the progressive rise in atmospheric oxygen. The Gunflint rocks, dated to about 1.9 to 1.888 billion years ago, offer a window into this pivotal period. At that time, phototrophic microorganisms capable of capturing light energy to feed themselves already played a key role in coastal ecosystems. In this ancient sea, complex microbial mats developed on the surface of the sediments, the result of associations of various microbes. Each occupies specific layers and adopts distinct strategies to survive. Within the Gunflint cherts, scientists have identified several emblematic fossil taxa. Among them, Gunflintia is particularly widespread and appears as very thin filaments, about 1 to 5 micrometers in width, with a micrometer equaling 1,000th of a millimeter. These filaments, invisible to the naked eye, have been observed through ultra-thin sections and powerful microscopes. Gunflintia could represent phototrophic bacteria, exploiting solar light to produce organic molecules that nourish the entire microecosystem. Another major group is Huroniospora. It appears in spherical or coccoid form with a diameter of 5 to 20 micrometers. These spheres, larger than the Gunflintia filaments, could correspond to individual cells or small compact colonies. They seem to have played a role in photosynthesis or other light-dependent processes. Indeed, they are found associated with the same laminar formations as other phototrophic microorganisms. Preservation by silica has maintained remarkable cellular details, offering a direct glimpse into the microscopic world of nearly 2 billion years ago. Among the observed forms, Eosphera is distinguished, characterized by large spherical conglomerates. These colonies sometimes exceeded several tens of micrometers, an already remarkable size for the fossil record of the time. Their organization suggests cooperation among microorganisms to exploit light and nutrients more effectively. Some researchers believe that Eosphera, thanks to these large structures, captured solar energy better in the clear waters of the Paleoproterozoic, while using dissolved iron for essential biochemical reactions. More rarely, the Gunflint deposits have revealed spiral shapes, much less frequent than Gunflintia, Huroniospora, or Eosphera, but just as remarkable. These fossils suggest a much broader morphological diversity than the described types. In a light-drenched environment, several phototrophic modes could coexist, each adapted to a niche. The spirals suggest a particular movement or strategy to better capture solar energy, reminding us that life in the Paleoproterozoic was far from uniform. To understand these organisms, one must examine the rapid silicification that preserved their membranes and cells. Silica-saturated waters penetrated the sediments and deposited fine crystals around and within the living structures before their complete decomposition. This early mineralization preserved many details, giving rise to chert. Without this process, most filaments and spheres would have disappeared. Experiments confirm that such encasement occurs in silica-rich waters, common in the Paleoproterozoic littoral due to volcanism and interactions with basaltic rocks. The chemical composition influenced the vertical distribution of microbial communities in the Gunflint. In these phototrophic mats, iron and sulfur gradients created, just below the illuminated zone, redox reactions. Some bacteria used the dissolved iron, others thrived in low-oxygen environments. This zoning favored species specialization and organic matter recycling. These conditions also contributed to the formation of iron deposits and the preservation of microbial traces. A key aspect of the Gunflint Biota is the organization of trophic networks. The organic matter from phototrophs fed other microbes, triggering strong reactions in the mats. This cycle led to the microbial phosphorus pump, which retained this element in biomass or minerals. This retention limited its dispersion in the sea, restrained productivity, but ensured an essential supply for DNA synthesis and guided microbial evolution. Between 1.9 and 1.7 billion years ago, the oceans remained largely iron-rich. The deep waters contained much dissolved iron, lacking sufficient oxygen to oxidize it. In some coastal areas, there was conversely photic euxinia, where the ocean's illuminated layer was saturated with hydrogen sulfide. These particular conditions profoundly marked marine ecosystems. Researchers have reconstructed these conditions through the chemistry of ancient sediments that preserve traces of bacterial activity. In low oxygen environments, dissolved iron circulated before precipitating under certain constraints. On continental margins, the abundance of organic matter and sulfate from erosion favored the production of sulfides by specialized bacteria. In the photic zone, there sometimes persisted a thin illuminated layer, but too laden with sulfides for normal oxygenic life. In this context, some microbial populations exploited the chemistry of the environment. Sulfur phototrophic bacteria called chlorobi performed an oxygen-free photosynthesis using hydrogen sulfide as an electron source. They thus occupied the suboxic zone, just below the surface. Higher up, cyanobacteria thrived thanks to light and locally produced oxygen, creating a fragile balance between two modes of phototrophic life. This superposition of phototrophic organisms was indicative of a fragile balance. Chlorobi occupied areas rich in sulfides, while cyanobacteria depended on light and oxygen. On poorly oxygenated continental shelves, microbes were distributed according to chemical gradients. At the bottom, some used iron or sulfates, while on the surface, oxygen production remained limited, thus sustainably maintaining ferruginous and sulfidic conditions. It is a dynamic that continues to intrigue researchers. A key factor in this configuration was the availability of phosphorus and molybdenum, two essential nutrients. Phosphorus was part of the composition of RNA and DNA, while molybdenum activated enzymes necessary for nitrogen fixation. When the latter was lacking, nitrate assimilation became difficult, favoring microorganisms capable of directly fixing denitrogen. Thus, their distribution influenced the productivity and balance of nitrogen metabolisms. Primary production, or the synthesis of organic matter through photosynthesis, was heavily dependent on access to nutrients. In iron-rich areas, the recycling of organic matter was ensured by various bacteria and archaea, using sulfides and sulfates to extract energy. At the same time, cyanobacterial activity could be limited by the lack of molybdenum, essential for nitrate reductase. These imbalances explain the durable coexistence of ferruginous conditions and relatively stable productivity. The balance between iron in the +2 state and hydrogen sulfide influenced the precipitation of minerals. In sediments, iron combined with sulfide to form pyrite. On a microscopic scale, this process could encapsulate cells or biofilms, generating fine pyritizations. Observed in core samples, they attest to microbial fossilization. Thus, iron and sulfur played a direct role in preserving ancient organisms. When phosphorus accumulated in bottom waters, another type of fossilization occurred, phosphatization. The earliest known phosphatized biofilms date from this period. They show that some microbial mats could trap the excess phosphorus. By mineralizing, these organisms and their secretions were preserved in the form of phosphate-rich films, offering researchers a unique window into ancient microbial diversity. The organic films preserved in these sediments reveal an intense microscopic life. Geologists analyze their size, shape, and arrangement to reconstruct ancient food chains. Sulfur phototrophic bacteria, cyanobacteria, and other microbes of the lower layers formed a complex network of exchanges. Dead cells served as a substrate for heterotrophs, and fermentation recycled the matter. The slow accumulation of minerals around this network favored its partial preservation. Studying these samples allows for the retracing of ancient chemical gradients. The simultaneous presence of sulfur compounds and ferruginous minerals suggests that coastal basins alternated between iron-rich bottoms and layers laden with hydrogen sulfide. In some cases, water circulation renewed the photic zone, favoring the development of cyanobacterial filaments. The combined analysis of minerals, organic matter, and fossils sheds light on the dynamics and evolution of these ancient ecosystems. Despite these advances, questions remain about the impact of ferruginous and sulfidic environments on the evolution of biodiversity. Some researchers believe that the limitation of oxygen delayed the expansion of complex life, while others suggest that local variations in nutrients and the coexistence of phototrophic and heterotrophic bacteria fostered adaptive innovations. Studying these rocks provides a unique insight into conditions before the rise of more oxygenated oceans and abundant multicellular life. Between 1.8 and 1.6 billion years ago, Earth went through a phase where new microbial actors appeared. Dominated by prokaryotes, communities saw the emergence of the first eukaryotic cells, with more elaborate internal structures, heralding future diversification. In shallow and sometimes warm seas, bordered by rocky platforms, microbial mats proliferated. Some organisms among them distinguished themselves by their size, their walls, and their ability to interact differently with their environment. These eukaryotic organisms are called cells, possessing a nucleus and often other internal compartments. Paleontologists distinguish them from bacteria and archaea through several criteria, including the morphotypes observed in sediments. Under the microscope, large rounded spheroids are sometimes identified, larger than most bacteria, with thick walls and sometimes ornaments evoking a complex internal structure. Their diameter, sometimes several tens of micrometers, reveals a higher level of organization. Besides the spheroids, some deposits reveal spiral ribbons called Grypania. Measuring more than a millimeter in width and several tens of millimeters in length, they far exceed the size of bacterial filaments. Their regular coiling suggests that they represent one of the oldest macroscopic forms of eukaryotes. These fossils indicate that the microscopic world was no longer limited to small cells, but already showed diverse forms, including large spiral ribbons. By examining these organisms, plausible steranes, molecules derived from sterols, are sometimes identified. In modern living organisms, their synthesis requires dioxygen, suggesting that their fossil presence may indicate eukaryotes. This implies that between 1.8 and 1.6 billion years ago, certain environments contained locally enough oxygen to support these biochemical pathways. Thus, even in an overall oxygen-poor atmosphere, microcommunities managed to create and maintain slightly oxidized niches. Linked to the steranes, the presence of internal structures like peroxisomes and mitochondria in these early eukaryotic cells is suspected. Peroxisomes detoxify certain oxidant byproducts, while mitochondria produce energy through aerobic respiration. Even if rocks don't allow direct observation, these proto-eukaryotes already seem to possess a cytoskeleton, internal membranes, and sophisticated mechanisms for managing energy, far more complex than in prokaryotes. Ecologically, these new eukaryotes are distinguished by their modes of nutrition. Osmotrophy allows them to directly absorb dissolved molecules, while phagotrophy involves engulfing particles or other microorganisms. Thanks to invaginations of the membrane-forming vacuoles, some small eukaryotes could digest bacteria. This mechanism, absent or rudimentary in most bacteria, marks one of the first forms of micropredation and opens up new ecological niches in the microscopic world. In these sediments, microbial mats of cyanobacteria testify to photosynthesis, forming viscous surfaces or mineralized column structures. The nascent eukaryotes coexisted with these communities, exploiting the available organic matter and developing survival strategies. One can imagine complex interactions. They benefited from the oxygen produced, while the micropredators modulated the density and composition of the mats. Even on a small scale, the ecosystem relied on exchanges of oxygen, nutrients, and organic molecules. Different geological formations provide valuable fossil evidence. Samples from the Roper Group in Australia offer a unique glimpse into this period. In these shallow marine layers, microfossils suggesting more complex organisms are found. The fluctuating conditions, nutrients, temperature, salinity, and day-night cycles created oxygen gradients. It is in these environments that primitive eukaryotes found various niches to adapt and diversify. At the other end of the globe, the Vindian rocks in India reveal similar clues. These deposits feature large spheroids, filaments more elaborate than bacteria, and complex microbial mats. Intriguing microstructures and traces of sterile synthesis are observed. The presence of these fossils on multiple continents suggests that these flourishing eukaryotes were widely distributed and not confined to a few local niches. In China, early formations also deliver comparable microfossils. Large, thick-walled spheroids suggest protective substances against physicochemical variations or microscopic predators. Ribbons reminiscent of Gripania also appear. The convergence of these discoveries across the globe reinforces the idea that the development of eukaryotes was not localized, but a global and progressive transition, shaped by the chemical and biological evolution of the planet during the Proterozoic. Beyond their structure, one can imagine the daily life of early eukaryotes. They lived in turbid and warm waters, sometimes partially dried on coastal margins. The mats of cyanobacteria provided the necessary oxygen, but just below, conditions could become anoxic. The eukaryotes thus positioned themselves at the edge of the mats, accessing nutrients and oxygen. This distribution, guided by microchemical gradients, favored their multiplication and the diversification of early food webs where phagotrophy played a key role. Micropredation, the ability to ingest other cells, becomes a pivot of microbial communities. Eukaryotes that master it obtain nutrients directly, without relying solely on dissolved substances. With the evolution of membrane invagination and intracellular digestion, they can consume bacteria and small eukaryotes. This innovation favors the complexity of trophic networks and initiates a cellular diversification that, hundreds of millions of years later, would give rise to life forms visible to the naked eye. The Mesoproterozoic is often called the boring billion, yet this period saw stable microbial productivity and the emergence of new eukaryotic forms. Coastal plateaus, bathed in weakly oxygenated waters, presented limited nutrients, notably phosphorus and certain trace elements. Despite these constraints, the ecosystems endured for hundreds of millions of years. The configuration of the continents and calm tectonics likely favored this astonishing biological and chemical stability. In these marine waters, life was based on microbial communities capable of withstanding changing conditions. Dissolved oxygen, albeit modest, allowed the establishment of photosynthetic organisms. The diversified cyanobacteria fixed carbon to produce organic matter and released oxygen locally. They coexisted with microeukaryotes, including acritarchs, forming a stable and productive ecological network despite globally low oxygen concentrations. Microbial mats extended over vast platforms, forming laminated structures by trapping and precipitating mineral particles. Mixed with clay or other fine sediments, they gave rise to stratified deposits, testifying to biological accumulation. The ecotones, transition zones between light and darkness, were stable over hundreds of millions of years, maintaining constant chemical gradients. The carbon cycle, sulfur circulation, and iron availability thus followed an integrated and regular functioning within the ecosystem. In this stable environment, acritarchs appear and diversify. Among them, Schweiuspheridium emerged as early as 1.6 billion years ago. Although microscopic to us, they far exceeded the size of bacteria. Their robust and extensible wall reflects environmental adaptations. The complex patterns, spherical or adorned with subtle protrusions, reveal strategies for protection against predators and mechanisms to maintain buoyancy in the water column, illustrating the ingenuity of these early microeukaryotes. Among the Mesoproterozoic acritarchs, Topania stands out with its large cells ranging from 30 to 160 micrometers and its tubular pseudopods. These membranous extensions allowed it to capture nutrients and explore its environment, or even move slightly within the microbial mats. Although their kinship remains uncertain, these protists illustrate a key milestone towards more complex eukaryotes and testify to a great ecological plasticity, interacting with other microorganisms for nourishment. Alongside the rise of eukaryotes, cyanobacteria remained central on coastal shelves. Some produced aconets, larger resting cells rich in internal reserves, capable of withstanding periods of cold or low light. The archaeoellipsoids fossils, ellipsoids measuring 10 to 30 micrometers, testify to this dormancy strategy. This capability illustrates microbial ingenuity and the resilience of communities throughout the Mesoproterozoic. Productivity seemed stable in the long term, but cyanobacteria and other producers nourished a complex network. Micropredatory eukaryotes, actors of the Protestant grazing revolution, consumed bacteria and microorganisms, accelerating the recycling of carbon and nutrients. This activity made the biogeochemical cycle more dynamic, enhancing resource retention and the continuous circulation of organic matter between niches. This resulted in a more resilient ecosystem and a more efficient biological pump. Despite its nickname of the boring billion, the Mesoproterozoic reveals a rich history of discrete diversification and metabolic innovations. The refinement of trophic interactions and the emergence of organisms with reinforced walls show that Earth was gradually setting the stage for the biological upheavals to come. Thus, this period laid the foundations for increasing complexity, ready to unfold when conditions became favorable for the rise of multicellular life. About 1 billion years ago, Bangiomorpha pubescens, one of the first multicellular red algae, appeared. This multicellular filament, probably attached to a substrate, marks the beginnings of multicellularity and sexual reproduction. Its study reveals how life became more complex, following deep geological and biological transformations on the planet. The name red algae comes from a characteristic pigment. It is phycoerythrin that colors these plants red. Modern red algae, grouped under Rhodophyta, often possess complex cell walls rich in carrageenans and agar. Bangiomorpha pubescens already shows some biological traits of this lineage, despite an ancient environment with low oxygen and distinct coastal chemistry, revealing the origins of these fascinating organisms. The multicellular filaments of Bangiomorpha pubescens illustrate a major transition from unicellular life to a structured organization. Multicellularity allowed for an efficient distribution of functions, better environmental resistance, and upward development to capture more light. The fossils reveal apical growth, where the algae developed from a specialized extremity, similar to the meristems of some modern plants. The fossils reveal structures suggesting meiosis, a process that produces gametes destined to merge and create a new individual. Before this, a sexual division dominated rapidly, but with little diversity. Meiosis combines the information of two cells, generating offspring better adapted to environmental challenges and paving the way for greater genetic diversity. The alternation of generations still emerging at that time illustrates the sexuality of Bangiomorpha pubescens. This cycle alternates between a haploid phase producing spores or gametes and a diploid phase developing a multicellular organism subject to meiosis. Although incomplete fossils do not allow a reconstruction of the entire cycle, the evolution of reproductive organs clearly shows the appearance of this complex mechanism, foreshadowing the reproductive strategies of modern red algae. Bangiomorpha pubescens is distinguished by its ability to cling to hard surfaces thanks to small specialized structures similar to crampons. These enabled it to attach to rocks, carbonate platforms, or stable fragments of organic matter. In shallow waters, subject to tides and currents, this fixation was an essential asset. The fossils show that these filaments colonized the photic zone, taking advantage of the available light. The photic zone corresponds to the part of the water illuminated by the sun, sufficient for photosynthesis, generally up to 200 meters depending on water clarity and the season. Bangiomorpha pubescens evolved in these shallow, light-filled and stable seas. Cyanobacteria proliferated on the same substrates, forming mixed microbial mats with the algae, testifying to early interactions between different phototrophic life forms. In microbial mats, photosynthetic bacteria, archaea, and small eukaryotes coexist. Each layer performs various biochemical processes, such as producing oxygen, decomposing organic matter, or fixing dinitrogen. Bangiomorpha pubescens occupied the upper stratum, exposed to light. Its interactions with cyanobacteria favored resource sharing, stabilized the substrate, and optimized nutrient recycling. The overall productivity of the ecosystem is thus enhanced. The appearance of Bangiomorpha pubescens increased the complexity of marine biofilms. Its multicellular filaments and advanced reproduction introduce new levels of organization, creating a microhabitat that offers niches to other organisms. Heterotrophic bacteria could settle there, consuming organic molecules released by the algae and participating in a food network still modest, but already more diversified than in previous microbial communities. The impact of Bangiomorpha pubescens on the nutrient cycle lies in its ability to reshape the distribution of elements. By photosynthesizing, it captured carbon dioxide to produce organic matter and release oxygen. Microbes broke down residues, returning phosphates and ammonium, feeding algae and other organisms. This positive feedback loop reinforced filament growth and the development of associated communities. This amplified the ecosystem's productivity. The study of Bangiomorpha pubescens fossils in the Baffin region reveals filaments with closely bonded cells and apical growth zones indicating cellular specialization. These fossil remains are very rare, as their preservation requires rapid burial, minimal disturbances, and geochemical processes capable of mineralizing the delicate tissues of red algae, providing a valuable insight into their morphology and organization. This discovery sheds light on the accelerated diversification of life at the end of the Mesoproterozoic. During the Tonian period, between 1 and 0.8 billion years ago, the microbial world of Earth changed profoundly. Unicellular eukaryotes diversified in the oceans, leading to the appearance of fossils known as acritarchs and novel micropredators. These new life forms energized marine ecosystems and prepared the emergence of more complex ecological systems. Acritarchs are microfossils with organic envelopes, generally associated with algae or other unicellular eukaryotes. During the Tonian period, they exhibited complex forms, sometimes equipped with spikes, bumps, or varied patterns. These features were likely not decorative, but served as defense against predators or facilitated floating. This increased their chances of survival in ancient oceans. Among the most remarkable Tonian acritarchs is Trachyhystricosphera imica. It is used as a biostratigraphic marker to date certain sedimentary layers. This fossil is distinguished by an envelope adorned with robust protuberances. Other genera, such as Valeria and Dictyosphera, also display elaborate ornamentations, revealing a great diversity of morphological adaptations. The organic envelopes of these acritarchs were so resilient that they have been preserved in sediments for millions of years. This robustness favored the preservation of their ornaments, which have become valuable benchmarks for tracing the evolution of micro-eukaryotes. The study of their morphology allows researchers to reconstruct some of the environmental and ecological pressures of that time. In parallel, the Tonian world saw the rise of more complex micropredators, considered primitive representatives of certain protists, such as amoebozoans and rosarians. They are known as vase-shaped microfossils with ovoid chambers and a neck-like opening. Due to this protective casing, they fed by phagotrophy, ingesting other microbes. This feeding strategy is essential to understanding the marine networks of the time, suggesting an intensification of interactions between different planktonic organisms. Phagotrophy, the direct absorption of organic particles, is distinct from the ingestion of dissolved nutrients. The Tonian micropredators thus captured bacteria and cyanobacteria, exerting pressure on these populations. With the multiplication of these hunters, competition for resources intensified. This favored the evolution of defensive strategies in prey. In this transforming environment, the recycling of phosphorus and other trace elements became crucial. By consuming nutrient-rich cells, micropredators released essential substances into the water for the growth of other organisms. This recycling allowed the Tonian biosphere to maintain stable productivity despite sometimes fluctuating conditions. Besides competition, certain cooperations existed between micro-eukaryotes and bacteria. The latter benefited from the metabolic byproducts of their partners. For example, a colony of nitrogen-fixing microbes provided nitrogen to neighboring eukaryotes, which in turn released organic compounds that nourished the bacteria. During the Tonian period, the concentration of oxygen in the surface ocean varied by region. This is referred to as oxygen pulsations. Oxygen pulsations denote episodes where oxygen temporarily increased in certain coastal or plateau areas. These fluctuations resulted from both the photosynthesis of cyanobacteria and other algae and the microbial respiration that consumed the oxygen. When oxygen increased, the oxidation of organic and mineral materials intensified. A higher oxygen level also favored the formation of nitrates from ammonium, thanks to nitrifying microorganisms. This enriched the water with nitrates and sometimes stimulated surges in planktonic productivity. This episodic increase in oxygen coincided with better availability of metals like copper and molybdenum, essential to the enzymes of nitrification. Copper facilitates the oxidation of nitrogen compounds, while molybdenum is involved in various reactions of the nitrogen cycle. When these trace elements were more accessible, nitrification became more efficient. The coexistence of primary producers, micropredators, and recyclers shaped dynamic marine ecosystems. Cyanobacteria and other unicellular algae produced organic matter through photosynthesis, while vase-shaped micropredators and other phagotrophic eukaryotes fed on these producers or on heterotrophic bacteria. The remnants and waste of this food chain were then decomposed by recycling microbes. This complex trophic structure created a feedback loop in which nutrient release stimulated new growth peaks. Some species of elaborately shaped acritarchs thrived in these favorable conditions, multiplying in diverse environments. The micropredators, by reinforcing selective pressure, pushed acritarchs and other micro-eukaryotes to develop innovative defense or reproductive strategies. Defensive adaptations included thicker shells, sharp ornaments, or repulsive substances. Acritarchs developing these strategies improved their chances of survival against micropredators, while the latter refined their ability to consume these protected cells. This ongoing evolutionary competition contributed to maintaining Tonian biodiversity. The rise of phagotrophy also influenced the evolution of reproduction in some micro-eukaryotes. The earliest forms of sexual reproduction may have developed to produce more diverse offspring, better adapted to predators. The appearance of cystic stages, allowing survival in unfavorable conditions, also fits into this adaptive dynamic. The Tonian thus proves to be extremely crucial for understanding how micro-eukaryotes established the foundations of an increasingly organized planktonic life, foreshadowing their future evolutionary successes. Between 800 and 720 million years before our era, Earth experiences a pivotal phase for microbial life. In shallow seas, microbial mats organize themselves into thin layers a few millimeters thick, a lit and oxygenated surface, and oxygen-free depths. These communities function like true microcities. At the top, photosynthetic cyanobacteria produce oxygen and organic matter. Lower down, where oxygen disappears, specialized bacteria, notably sulfate-reducing ones, recycle the matter using sulfur, iron, and carbon. Small channels diffuse gases and nutrients into the heart of the mat, and tiny carbonate granules form and cement the structure. At the landscape scale, this microbial activity deposits carbonates and builds sedimentary structures, thrombolites with lumpy texture, and laminated stromatolites, sometimes in columns. In some places, eukaryotic grazers carve the surface and leave small cavities. These archives keep the record of the chemistry of the time. Measurements of carbon-13 indicate variations in productivity and carbon burial. Those of sulfur-34 track the intensity of sulfate reduction. Together, these clues narrate how these mats influence the carbon and sulfur cycles and, more broadly, the composition of the oceans. As long as the climate remains mild and nutrients available, these communities thrive in vast sheets on the marine platforms. Then a major disruption occurs. The Sturtian, during the Cryogenian, an episode of Snowball Earth. For several tens of millions of years, ice caps stretch to low latitudes. Oceans are often trapped under ice, but life clings on in refuges. Polynius, Ice-free openings, especially towards the equator, let in some light. On the ice surface, cryoconites, small melted pockets, enriched with dust and organic matter, host cycrotolerant cyanobacteria and other microbes protected by biofilms. Under the ice, where rock keeps the temperature above zero, circulate subglacial waters, pour in light. There, chemolithotrophic archaea and bacteria derive their energy from mineral compounds, iron, sulfur, without relying on the sun. On oceanic ridges, hot hydrothermal vents provide heat and minerals. Around the smokers, specialized microbial communities thrive, independent of photosynthesis. Subglacial lakes, comparable to today's Lake Vostok, constitute other refuges, dark, oxygen-poor, but nutritionally supported by mineral and volcanic fluxes filtering through the ice. Even residual light plays a role. Photoferrotrophic microbes, capable of using ferrous iron as an electron donor, exploit the weak radiation passing through the ice sheet. They precipitate iron oxides, sometimes leaving characteristic geological signatures. Overall, these extreme ecosystems rely on protective biofilms, matrices of exopolymers where bacteria and archaea huddle together, exchange nutrients, and defend against frost. Volcanism, by releasing iron, phosphorus, and silicon, triggers spasmodic pulses of nutrients that locally rejuvenate biological activity. After the Sturtian, between 659 and 651 million years, a climatic respite sets in. Ice recedes in places, melting loads rivers with sediments and nutrients, notably phosphorus. In coastal areas, cyanobacteria and unicellular algae explode in blooms. The microbial loop intensifies. Primary producers convert light into biomass. Phagotrophs ingest this biomass. Other microbes decompose waste and recycle nutrients. In some coastal pockets, oxygen becomes more present. Enrichments in elements sensitive to oxidation, molybdenum, uranium, bear witness to it. This oxygenation remains heterogeneous and intermittent, but it suffices to diversify respiration modes and stimulate nitrogen fixation by certain cyanobacteria. Then comes the second great cryogenian glaciation, the Marinoan, lasting until 635 million years ago, marked by rapid back and forth between freezing and thawing. The ecological pressure is strong. Light is scarce under the ice. Nutrients are trapped. Temperatures are unstable. Microbes activate survival strategies. Many enter dormancy as thick-walled organic cysts, capable of resisting freezing and chemical shocks. Others reinforce their walls with biopolymers that retain water and filter toxins. Some partially silicify, mineralizing their envelope to better withstand salinity and temperature variations. Mucilaginous matrices thicken biofilms and cushion stress. Cooperation becomes an asset. Within mutualistic consortia, cyanobacteria and heterotrophs exchange resources and vitamins, improving collective resilience. Productivity remains low, concentrated in the rare polynyas where photosynthesis is still possible and where very simple trophic chains persist. At the end of this glaciation, the global ice sheet collapses in a short time period on a geological scale. The deglaciation releases sediments and nutrients, reorganizes winds and currents, and triggers upwellings of deep waters rich in nitrogen and minerals. Microbial productivity picks up again. On shallow seabeds, laminated microbialites stack layer upon layer, true archives of the transition, observed today in Africa, Australia, and North America. Eukaryotes, algae and protists, diversify once more. Acritarchs experience a revival of diversity in forms. Predatory protists equipped with cilia or pseudopods hunt bacteria and other eukaryotes, intensifying selective pressure. Locally, coastal waters become more oxygenated, favoring more efficient aerobic respiration and preparing the rise of more complex biological systems. The climate gradually warms. Ocean circulation becomes more dynamic, distributing nutrients and oxidants and stabilizing various chemical gradients. Basins fill with clays, quartz, and carbonates, and microbe carbonate structures consolidate. Everything is in place for the later rise of Ediacaran forms. In parallel to this climatic storyline, it is essential to understand the functional anatomy of a microbial mat, for it traverses all these periods. On the surface, the photic zone captures light. Cyanobacteria fix CO2, release O2, produce exopolymers that retain water, trap particles, and stabilize the structure. Some also fix atmospheric nitrogen into assimilable compounds. Just below, a transitional zone combines the last traces of oxygen with other metabolic pathways, nitrate utilization, iron and manganese oxidation or reduction, exploitation of decomposing organic matter. Even lower, the sulfidic base is the realm of sulfate reducers. They convert sulfate into hydrogen sulfide, reactive and toxic to many, but integrated into the local sulfur cycle through its reactions with dissolved iron and the formation of sulfides. Photoferrotrophs often occupy the shaded layers. They oxidize ferrous iron using low light, precipitating iron oxides. These communities are not static. They migrate on a millimeter scale in response to light. Some bacteria ascend, others descend. These movements create small domes, trap particles, and exopolymers, and build over time successive laminations, the recipe for stromatolites. In cross-section, the alternation of light and dark layers reveals different phases of growth and sedimentation, recording the distribution of metabolic groups according to local light and chemistry. At each stage, the microbes adapt. They build carbonated landscapes, shape chemical cycles, invent protections, exchange genes, and interact with new predators. These innovations, patiently shaped in changing environments, prepare the great transition to more oxygenated, more structured ecosystems, and soon inhabited by life forms visible to the naked eye. It is this long microbial acclimatization, inscribed in stone and in the chemistry of sediments, that made possible the subsequent history of life.